Welcome to Biology B. This recording is in addition to our live class session that we had today on January 4th, 2017. We've been talking about bacteria, and the last item that we did in class was to fill in this chart. So make sure that you know the definitions of each of these terms and you're able to fill in this chart. So let's move on to the items that we did not cover in class but are part of your reading for today. Okay, so we talked a little bit about bacteria being able to function with oxygen or without oxygen, but now we can give that an actual name. So bacteria can use the process of fermentation to obtain energy when oxygen is not available. So instead of using cellular respiration, it can actually use, or photosynthesis, it can actually use fermentation. And the bacteria that uses this process is called an obligate anaerobe. So it really does not like oxygen. It's a hating oxygen bacteria, or oxygen-hating bacteria. Next, we have the bacteria that needs oxygen to obtain energy for cellular respiration, and that they are called obligate anaerobes. Or, I'm sorry, aerobes, not anaerobes. So obligate aerobes. They are oxygen-loving. And then we have facultative anaerobes, and they use both oxygen and fermentation to break down their nutrients into a usable form. So all the structures and functions we have been discussing up to this point are considered adaptations for survival. So survival of the fittest, the organism wants to be able to survive, wants to be able to produce, reproduce, and carry on its DNA into a new generation. Another adaptation that makes bacteria really unique is this ability to create an endospore. And that is a coat that surrounds the bacteria to protect it when the environment is harsh. It protects the chromosomes within the cell and a small part of the cytoplasm. But the cell dies, and then when the conditions are better, then it actually will come back to life. And it can survive over long periods of time in very extreme conditions. So I want you to think about where we can think about Mars. And one of the things that the Mars team, so the team of scientists that is studying Mars, one of the things that they're looking at is, are there resources there or items that living organisms need in order to survive? Or is it possible that there was actually life on Mars at one point? So the rover, the Mars rover, is moving around on the planet looking at different samples, trying to see if there's a possibility that it might have lived on Mars. And we might even uncover an endospore or something like it. So that's one thing that the rover is doing. We have not found any remnants of life, so no fossil record of life on Mars yet, but we do know that the elements that life requires exist on Mars, but the conditions are very extreme. Okay, next. So mutations within the bacteria. We spoke about conjugation, which is a part of the reproduction of bacteria where it moves genetic material from one bacterial cell to another using the pili. That process, adding in that genetic material can create a mutation, and there can also be random errors during binary fission that can create mutation, but it basically is a change in the DNA sequence. And this can lead to new genes, new combinations, new characteristics, and genetic diversity. So bacteria reproduce very quickly, and some every 24 hours, some even faster than that. And because of the mutation, we see a change quickly, so we can monitor evolution within bacteria very quickly. So one of the questions to think about is why it is important that bacteria have diversity. Why would that be so important? Well, if the environment changes, some bacteria might not have the right combination of genes that will enable it to survive and repopulate. So that would mean that it would become extinct. But because of diversity, the bacteria is able to live longer and have reproduction to pass on its genetic material. An example of this is when antibiotics are introduced to the system. If you don't take the antibiotics as prescribed, some of the bacteria may live. So some of the bacteria that has traits that allow it to live might survive. And then it's going to carry on that genetic trait onto its, daughter cells. An example of this is antibiotic resistance. And this happens from the special outer layer, polysaccharide layer, that we find on bacteria. There are some bacteria that that layer is larger than others and harder for the bacteria to get through, or it's not, this doesn't have the same charge on the outside layer of the bacteria, so the antibiotic won't stick to it. Oops. So how do bacteria benefit plants and humans? They're not always harmful. Bacteria are decomposers, so they break down our waste. They break down dead organisms and put it into soil to make the soil nutrient rich. They get energy from dead organisms and return those nutrients back into the environment. Most do not cause disease. They can be very helpful. And here's a couple examples. So benefits to plants. All organisms require nitrogen to make proteins and DNA and RNA. So most nitrogen is in a gas state. It's in our atmosphere. And it's not in a form that plants can use. So nitrogen can be put into a usable form through lightning, but we don't see a lot of lightning strikes, right? It's not happening all the time, and so it doesn't really put in enough nitrates, usable nitrogen, into the soil for plants. And so bacteria lives in the soil and breaks down the organic material, and also can pull in the gas and add in hydrogen to it that makes it into a usable form. So we have nitrates that are NH2 or NH3, and NH3 is the usable form of nitrogen for plants. Now, plants have a special adaptation that we're going to learn about when we do our dissection. And close to the end of this trimester, so like this first, I want to say the end of the first week of February, because the last day of the trimester is on February 15th. So that week we're going to dissect our plants that you guys are growing now, and if you haven't planted it, attend the lab lesson tomorrow, and we'll on the 5th, and I'll go over how to plant those. But we're going to talk more about the adaptation and the ability for bacteria to help the plant through this nitrogen fixation process. There are benefits to humans. For example, your body is covered in bacteria, which is called normal flora. This flora helps prevent human bacteria from infecting your body and causing disease. An example of this is E. coli. So E. coli is in our environment, all over our body, but it can make vitamin K, and our bodies absorb, our skin absorbs that vitamin K. And humans use vitamin K to help with blood clotting. So our platelets really require that vitamin K to function appropriately. There are some vitamin pills that are made with the help of bacteria. We have bacteria in our stomach that help us break down food, help us absorb vitamins, things like that. So we want the healthy bacteria in our stomach. Probiotics are listed as healthy bacteria. So if you take an antibiotic that destroys all the bacteria in your stomach, then you can take probiotics and it'll help put that good bacteria back in there. There is another benefit to humans. They've been looking at bacteria that eats radioactive material, bacteria that leaves a petroleum-based product that can be used as fuel. We use bacteria in our water treatment and our trash facilities to break down bad stuff. So we do use those. Well, not bad stuff, but break down the organic material. So those are some ways that bacteria benefits humans. But how does bacteria cause disease? A very small percentage of bacteria can cause disease, and it does it in two ways. So some bacteria multiply at an infection site and can spread to other parts of the body, and it creates an immune response. And so the symptoms that you feel or not feeling well, it really has to do with a decrease in your red blood cells because there's an increase in your white blood cells. So you get tired because of that. You can have a sore throat because the area where the bacteria is working or living and growing is taking resources and maybe destroying cells. And so that is one way that bacteria can cause disease in the body. Another way is that it can secrete a toxin or other substances, such an acid that can cause tooth decay. So that's an example. Have you ever had an infection that needed antibiotics? I know we discussed a little bit today strep when we were discussing the different shapes of bacteria. So you might have had just a fever, not feeling good, tired, and those are all symptoms of an infection, but it's possible that they were viral, which is treated differently than a bacterial infection. So some of the human diseases that are bacterial include STDs, so sexually transmitted diseases such as syphilis, gonorrhea, chlamydia. There are respiratory diseases like strep throat, pneumonia, whooping cough, anthrax. So these are all respiratory diseases and require antibiotics. Skin diseases such as acne, boils, infection of wounds or burns. We talked a little bit about staphylococcus today. Digestive tract diseases such as gastroenteritis, many types of food poisoning, cholera. Gastroenteritis is inflammation of the lining of your stomach, and it can cause you to throw up and not feel well. Food poisoning can do the same thing. Nervous system diseases, botulism, tetanus, bacterial meningitis, all are bacteria that impact the nervous system. These can secrete toxins that can break down parts of the nervous system. Okay. So next we're going to talk about viruses. A virus is a non-living strand of genetic material with a protein coat. It's not considered to be living because it does not meet all of the characteristics of life. So we talked about the characteristics of life in the first biology class, Biology A or semester 1. Characteristics of life consist of metabolism, ability to respond to its environment, homeostasis, has to be single-celled, or has to have a cell or cells, and in this case, and reproduce. So in this case, a virus doesn't have a cell. It does not necessarily respond to its environment, although that's argued. It does not have a metabolism, but it can hijack a cell to have. So it really doesn't meet the characteristics of life. Viral diseases cannot be treated with antibiotics. They can be treated with vaccinations. There is antiviral medication, and that just boosts your immune system to respond to the virus. So what are some viruses that you've heard of before? Maybe like chickenpox, herpes that you get on your mouth, like the herpes cold sore. Some of the human diseases caused by viruses are sexually transmitted diseases like HIV and genital herpes. I also spoke about cold sores. They're not sexually transmitted though. Well, I guess they can be, but they're a different form of the virus. Childhood diseases such as measles, mumps, chickenpox. We can get the varicella vaccine, which is a vaccine for chickenpox. Respiratory diseases such as common cold, influenza. Skin diseases like warts and shingles. So shingles can happen like chickenpox, and they have related shingles to individuals who have had a light case of chickenpox or not had chickenpox at all when they're an adult. They might have shingles. There are viruses that cause digestive tract diseases, so like gastroenteritis. So gastroenteritis can be caused from bacteria or a virus. And so doctors will give you a few days to see if it clears on its own, because our immune system can usually clear it before they give you antibiotics. There are nervous system diseases like polio, viral meningitis, rabies. Polio in the United States has been eradicated for the most part. Third world countries that don't have access to vaccines, many of these things like measles and mumps and polio and smallpox have not been eradicated. So it's one of the reasons why we monitor our border, and we have the CDC. So CDC monitors these things. There are other diseases like hepatitis and smallpox that we monitor closely. The origin of viruses is not known. So one theory, however, is that the virus came from parts of the cell. And scientists found that viruses are similar to genes in cells, and these genes somehow become able to exist outside the cell. Here we have the structures of the different types of bacteria. And they pretty much have all the same thing. One of the things that they have that are in common is the capsid. And the capsid is the outer layer made of proteins. So take a look at these. You can put them in your notes, but each one has a capsid. Each part of the virus, it has a special function. So its structure is related to its function. There are some different types of viruses. We have the adenovirus, and it causes a cold. We have the influenza virus. It causes the flu. A bacteriophage is a virus that infects bacteria. But notice that all three viruses have the same basic structure. They have that genetic material in the inner core, and they have a capsid that protects that genetic material. So we have the possibility of having DNA or RNA inside that capsid. And it can be classified as either a DNA virus or an RNA virus. Smallpox is an example of a DNA virus. So smallpox caused by DNA virus infected humans for thousands of years. And a successful worldwide vaccination program has pretty much eliminated the disease. So how does the virus actually infect its host? Well, it attaches to the host cell using receptors on the plasma membrane of the host. So it matches up to that. And the different species have receptors for different types of viruses. We also have antibodies. So if we have an antibody, then the virus can't necessarily connect to the cell. But for those cells that don't have an antibody, it can result in the virus passing on its genetic information into that cell. So once it's attached, the genetic material enters the cytoplasm of the host cell, and the virus then replicates by either the lytic cycle or the lysogenic cycle, and we're going to talk about both of those. So the lytic cycle is the active part of the life cycle of a virus, or the cycle of a virus. It starts by the host cell. Once it's been introduced into the host cell makes copies of the viral RNA and DNA. The host cell then makes a protein coat that encloses the RNA and DNA. And through exocytosis, so that's using one of the functions in the cell to push that information or that virus back out of the cell so it can infect other cells. Another way that it can cause those capsids or those viruses to be released outside of the cell is when the cell bursts. So the breakdown of the cell wall, and this is called lysis. Then the virus is free to infect more cells at this point. So one little virus can attach itself to a cell and reproduce very quickly. So it hijacks the cell processes, reproduces those viruses, and then lets them out so that they can go through the bloodstream, go through the cytoplasm and or the plasma in your body and attack other cells to do the same thing. Let's see. The next is the lysogenic cycle, and in this cycle, the viral DNA enters the nucleus of the host cell. The viral genes become a permanent part of the host chromosome, so it's already there. And when the cell divides, the virus is copied. This may stay inactive for months or years until something activates them. Different viruses are activated by different things. So when activated, the viral gene replicates by the lytic cycle and causes an active infection. So here we see an image of these two processes. So we'll start at the top. Oh wait, no, we'll start over here on the left where we have the entry. So the viral DNA enters the bacterial cell. And then it attaches, so the virus attaches to the bacterial cell, enters the bacterial cell, drops off its um, chromosomes, right? It pushes those chromosomes in there, and then the it can either hijack the processes in the cell, or it can just become part of the DNA in the cell. So in the lytic cycle, it just hijacks things and it starts producing more DNA or more RNA viruses, and it's an assembly line. The cells make the capsids to protect it, and then it's released out into the bloodstream to go infect more cells. So that's the lytic cycle. The lysogenic cycle is just that it is there in the DNA, and then when the bacteria splits apart through binary fission, then we end up with two cells with the same identical DNA structure. So that means that the virus that was input into the DNA has essentially changed the daughter cells. Cancer is created this way. So a virus hijacks the cell. The cells aren't able to regulate, and they keep splitting and splitting and splitting and splitting based on the DNA that's in the cell. Some viruses are made of RNA instead of DNA, and we call the RNA viruses a retrovirus. So it's just a type of RNA virus with a replication cycle that has many parts. The human immunodeficiency virus, also called HIV, is a retrovirus. So the HIV structure is similar to other viruses. It has a protein capsid, has RNA and an enzyme called reverse transcriptase. And the enzyme is really important because it's what makes DNA from the viral RNA. So it goes into the cell, it uses the things that are in the cell, and it creates DNA. HIV attaches to a host cell. So these are the steps. HIV attaches to a host cell and releases the RNA into the cell. Reverse transcriptase 3 makes DNA from the RNA, and the DNA moves to the nucleus of the cell to become part of the chromosome. And it might stay inactive for years. Once activated, the virus makes RNA from the viral DNA. So the cell has been officially hijacked. The host cell then makes new HIV particles instead of something else. And this is where we get into problems with HIV. Once it becomes active, we see a change in the immune system, specifically the C4 immune cell, or part of the immune system. And it makes it to where the cell does not, or T4, I'm sorry, makes it to where the human cell does not continue to create T4 cells. So we're eliminating the immune system. So that's what an active HIV, also called AIDS, is. So another piece of the puzzle is prions. And prions, there's also discussion about whether or not prions are considered to be alive, but we do classify them just like we classify viruses. They are a protein, so just like a small snipping of protein that really don't have a specific function in a cell, but they're floating around, and they can cause infection or disease. They normally exist in all cells. They're shaped like a coil, and they have mutations in genes, and this can cause these proteins to be created if there's mutations. Mutated prions can cause normal proteins to mutate. So the body might recognize them as an enzyme, but it's a mutated enzyme. And an example of a disease that's caused by prions is mad cow disease. So abnormal prions infect and burst nerve cells in the brain, leaving spaces that make the brain look like a sponge. And we saw this occur in England and when, sorry, when um, we get our meat from the grocery store, while the FDA monitors the herds of cattle or pigs or sheep, and they test for mad cow disease. So if a herd has mad cow disease, then they will kill that entire herd and they won't sell the meat. So that's what the FDA does. They look for that to help protect us. Can be devastating to a rancher, though. So I just want you to think about how you're feeling about what you've learned with regards to bacteria and viruses and prions, and make sure and utilize the worksheet that's in your reading. Review your reading, take notes, and then you should be prepared to take the quiz that is due on the 5th. If you have any questions, see me in Science Help. And this concludes our recording on bacteria and viruses.